Hey everybody, we're just going to run through these problems real fast and give an explanation on each one of them so we can help you all learn. That's the best way to do it. So for number one, I wouldn't take my explanation, I would take Sister Mayo's. Hers is a lot more clear. But basically, you want to take the normal chromosome and then the inversion chromosome and duplicate them, because that's what happens. Basically, we go through all of meiosis for each one of these chromosomes to get the answers. So if you do that, you're going to do L, M, N, chromosome or centromere, O, P, Q, R, S, T, Q, R, S, T, Q, V, W, X, U, V, W, X. And you do that again, L, M, N, O, P, Q, R, S, T, U, V, W, X. So you do that because it's been duplicated, and then you take the next one, and you want to look at the problem, what it's asking. It's doing between the T and the S. So you know it's going to be through this spot right here. So you want to line up the other ones right there. So you're going to do S, T. So if that's going to happen, the S is going to go R, W, X, and then the other way would be U, V, Q, P, U, V, Q, P, O, centromere, N, M, L. And then you just duplicate it. I'm going to take that line that was really poorly drawn the first time and go all the way down. So when you're doing that, to get the crossover, because it says a crossover between S and T, this one and this one stay the same. So that's going to be your answers for one of them. And then the other one, you're going to take it and it's going to be, did I do that right? I think so. You take it and you're going to, you cross over. I did it right, never mind. Yeah, you cross over, so you're going to take it. This one is going to connect with these one, and this is going to connect with this one. So the other one would be L, M, N, O, P, Q, R, S. Then it crosses over, so T, U, V, Q, P, O, centromere, N, M, L. And then for the next one, you're going to do X, W, R, S, T, U, V, W, X. So those would be your four. The two circled ones are just the same because there's not touching, so there can't be any crossing over. And then the other ones, this one would go with that one, and then this one goes with that one, and that's going to be your four. So that one, that one, that one, and that one. And that's your answer for number one.
Okay, so for number two, to fill in the blanks, question A asks, non-disjunction in what produces 50% aneuploid gametes, whereas non-disjunction in something else can produce a mosaic phenotype. And the first one is meiosis 2. The second one is mitosis. For B, it says meiosis in an individual who has suffered and the blank is paracentric inversion, produces a dicentric chromosome and an acentric chromosome. For C, Turner syndrome is an example of a human disease caused by monosomy, while familial Down syndrome is an example of a disease caused by a Robertsonian translocation. For D, the human cells that can be 4N, 8N, or 16N are liver cells. E, a terminal deletion in chromosome 5 causes, and that's the Cri-du-Chat syndrome, while the inversion in the X chromosome causes type A hemophilia. For F, a duplication is a change in chromosome structure that can be potentially beneficial to an organism or an entire species. And G, when a translocation cross is formed, the alternate segregation pattern can produce a viable gamete and offspring. And for number 3, these are questions concerning defects in chromosomes with their number and their structure. And so question A asks for these different syndromes, what are the total number of chromosomes that would be found in the human and have the following conditions. So for Turner syndrome, there's one less than normal, so that's 45 total chromosomes. For Jacobs syndrome, there's one more, so there's going to be 47. Cri-du-Chat syndrome does not actually change the number of chromosomes. And so that's going to give us 46, as well as familial Down syndrome, which is also 46, as well as type A hemophilia. So those are all going to be 46 with the normal number. For B, they ask which of the following terms is not associated with Charcot-Marie tooth disease, type 1A. So going through this, the ones that are not associated with it are triploid, aneuploid, reciprocal translocation, trisomy, and non-disjunction. So the things that do have to do with it are diploid and duplication. For C, it asks what are the following terms associated with Klinefelter syndrome. The following ones that are associated are aneuploidy, trisomy, and non-disjunction, which means the other ones don't have to do with it. And for D, which of the following are associated with type A hemophilia? It'd be none of these answers right here. None of them are affiliated with it.
All right, so for this next one, it's pretty long and difficult. Let me erase this. The question says, I'm just going to read it. It says, one pair of homologous chromosomes in a mouse has the following sequence of alleles, gives a sequence, and another pair of homologous chromosomes has the following sequence of alleles, gives that allele sequence. Suppose a reciprocal translocation occurs between one chromosome and each pair, producing the following allele arrangement on two of the four chromosomes, gives the allele on one of the chromosomes and another arrangement on the other. Note that there's still one chromosome with this, and then another chromosome with a different one. The star indicates the centromere. So the first one's just draw the diagram. So a good diagram is one in the book, or not in the book, in the PowerPoint. And I thought that was really well to show out the way that it's done. It doesn't have the letters to draw out which ones. So we just put in the one from the diagram on the PowerPoint because it was a lot to draw, and then we did followed the steps for that to get the answers for B, C, and D. So for that, I'm going to zoom in on our picture to kind of explain. So it says identify the allele composition of the gametes produced by the alternate segregation. So alternate segregation is, the answers would be two of them. So you can get four gametes because of mouse, you get four. So you can get two that are MNOP. I can just write it out. MNOP with the centromere, and then QRST, and then DTF. GH, the centromere, and then IJKL. And the way to do that is you're just going to look at the chart and follow it through by doing the same method that we did for number one. And then so you get two of those. Then you're also going to get two of D, E, F, P with the centromere, Q, R, S, G. And then another one with M, N, O, GH, IJKL. So you're going to get one for B. You're going to get those two arrangements just by simply following through the steps on the PowerPoint. And there's it's a really good chart you can just follow through. So for the next one, you can do very similar. You're going to get this same one right here for one, but it's going to be the opposite as the other. So MNOGHIJKL. So it's very similar to the ones that you did for that one, but the difference between an adjacent one segregation and a non-disjunction, or not non-disjunction, alternate segregation is just that. So these two would switch. And then the other one would make it the same. So DE, F, GH, the centromere, IJKL, and then D, E, F, P, Q, R, S. So these two just switch, and it just depends on the way that the chromosomes were going to cross. And then for the last one, for adjacent two, it's going to be very similar. So this one's going to stay the same, and this one's going to stay the same, but these two are going to switch. So that's going to be the biggest difference. And it just depends on where the chromosomes line up. And that's the main difference between adjacent 1 segregation, adjacent 2 segregation, and allele separation. I can never remember the name of it. I think that's what it's called. Allele segregation, alternate segregation. So that's the main difference, is just depending on where the alleles are lining up on the chromosomes during meiosis. And like we said, in the PowerPoint, there's a really good slide for that.
Okay, so for question 5, it says cats have 38 total chromosomes. How many chromosomes will be found in the following types of cats? So triploid, you take the haploid of 38, so half of 38, and you multiply that by 3 for a triploid cat. So we're going to get 57 for that one. 3n. So
what is that? 29? 19?
19, yeah. And then 19 times 3. And that gives us 57. For B, it says a cat that is monosomic for chromosome 12. And so of the 38 total, we only have one instead of 2 copies of it. So that gives us 37 instead of 38. So 38 minus 1. For C, a cat that has suffered a terminal deletion on chromosome 7 still leaves us with 38 for the cat. D, a cat that has suffered a Robertsonian translocation between 17 and 18 gives us 37, and again, that'd be 38 minus 1. And E, a cat that has suffered a reciprocal translocation between chromosomes 6 and 9. We don't actually lose any chromosomes, and so the total is still 38 for that one. And that is week 8, group 8, I think.
I don't know what group we are. We're your favorite group, Sister Mayo. I know it. Have a good week, everybody.